Well, so we're coming now to the really exciting part of the evening, which is the presentation by our Jak Kelly Award winner and the three winners of the scholarships. And I will, in a moment, be handing over to Dr. Osman to introduce the Jak Kelly Award and winners. Before I do this, I would say just a little bit about Jak Kelly Award. So it's or perhaps more about Jak Kelly. Jak Kelly was professor at the University of New South Wales in the School of Physics, and I see that he left in 1989. I knew him. He was a friend of mine, and he was a really fine man, a lovely man, humorous, good-looking, well-spoken, and very articulate. And I remember him telling me about science. I'm something of a physicist myself, and he told me about sputtering, a process by which you shoot. I only know this because he told me that heavy ions, charged atoms, you could say, I mean, are fired at high speed into a surface to do things. And Matthew is going to talk about a much more sophisticated, much more modern version of a little the same thing. Jak Kelly was also president of the Royal Society, of this Royal Society, for 2 years, and he is sadly no longer with us, but very much remembered. Well, so let me now introduce Dr. Fred Osman. Are you there, Fred, somewhere out there in virtual land? Fred Osman is here to represent the Australian Institute of Physics, because it is the Jak Kelly Award is the joint activity of the Royal Society and the Australian Institute of Physics. So over to you, Fred.
Thank you very much, and very nice everyone this evening for the Society's Jak Kelly Award and the Royal Society of New South Wales Scholarships for 2020. Professor Ian Sloan, the Royal Society of New South Wales President, members of the Royal Society of New South Wales, and distinguished guests joining us this evening for our 2020 Student Award presentations. For the past 11 years, the Australian Institute of Physics and the Royal Society of New South Wales has named a separate award category within the Australian Institute of Physics New South Wales Postgraduate Awards event called the Jak Kelly Award. As Ian mentioned, the Jak Kelly Award was created in honour of Professor Jak Kelly from 1928 to 2012, sadly died in 2012, who was the head of physics at the University of New South Wales from 1985 to 1989. And was made an honorary professor of the University of Sydney in 2004, and was the president of the Royal Society of New South Wales in 2005 and 2006. Professor Jak Kelly was appointed as an inaugural fellow of the Royal Society of New South Wales for his work of international stature in applied physics, and for his contributions to science generally and to the Royal Society of New South Wales in particular. The Jak Kelly Award was created to encourage excellence in postgraduate research in physics. It is supported by the Royal Society of New South Wales and the Australian Institute of Physics. The winner is selected each year from a short list of candidates who have made presentations at the Australian Institute of Physics Postgraduate Award event, where candidates are judged on the criteria: 1, content and scientific quality; 2, clarity; and 3, presentation skills. We were very fortunate to actually hold the event in person late last year for the Jak Kelly Award to actually determine our winner this year for Matthew Donnelly. The Australian Institute of Physics and the Royal Society of New South Wales has been honoured to award the Jak Kelly Award each year, and we are delighted to have this award presented to encourage excellence in physics as an important funded prize by the Society, to recognise outstanding achievements by early career individual working in a physics-related field. We would like to congratulate on this occasion Mr. Matthew Donnelly, PhD candidate from the University of New South Wales, who was selected as the 2020 Jak Kelly Award Winner for his presentation on controlling how electrons move in silicon at the atomic scale. Thank you very much.
Good evening, everyone. My name is Matt Donnelly, and I'm a PhD student in the group of Michelle Simmons at UNSW. And it's my pleasure to be here tonight and to give this talk. I wanted to first begin by thanking Professor Ian Sloan and Fred Osman, as well as the other members of the Royal Society of New South Wales who've made tonight possible. And so let's begin. So my talk is going to be entitled Controlling How Electrons Move in Silicon at the Atomic Scale, which is going to be based on the work that I've done in my PhD here at UNSW. The talk is going to be in 3 parts. In part number 1, I'm going to talk about electrons and condensed matter and introduce a couple of concepts that will be important throughout the talk. In part 2, I'm going to talk about tunnel junctions, which forms the bulk of the work that I've done in my PhD. And then in part 3, I'm going to talk about quantum computing and how my work fits into that field. But starting with electrons and condensed matter. To build up the picture of how electrons behave in condensed matter, we should first consider the simplest system that electrons exist in, which is an atom. So this model on the left here is of the Bohr atom, which is kind of the model of an atom that most people are familiar with. You have the protons and the neutrons in the nucleus there surrounded by electrons in orbit around it. The problem is that this model is quite unphysical, primarily because if electrons were to exist in orbits like this, they would radiate energy and eventually collide with the nucleus, which obviously does not happen. So what physicists needed to do is revise this model and come up with a model that is more realistic in its predictions. And so what happened is that the physicists eventually got to the model of the electron wave function, which we use to describe how electrons exist in atoms today. So the concept of a wave function is a quantum phenomena that is quite deep and quite difficult in a lot of ways to understand. But the simplest way to kind of come at it is that the wave function essentially does away with the concept of the electron existing as a single particle in a single place and introduces the element of probability. So these areas you see here in different colors on the plot on the right, they indicate different regions around the nucleus where electrons are more or less likely to be found, which is quite a conceptual jump, but is actually a lot more physically accurate according to our understanding at the moment. And these wave functions also describe the energy of the electrons around the nucleus. And this concept of electrons having a certain energy or an energy level is going to be important as we move forward. So moving on from this concept of an electron wave function in an atom, what happens when you have lots of atoms together? And that's what we consider in condensed matter. So condensed matter physics is where we talk about materials or matter where there are strong interactions between particles in that material. You can have solids, you can have liquids, or you can have more exotic phases of matter where this occurs. But today we're going to talk about primarily a solid, specifically silicon. And so this picture on the left here is of the unit cell of a silicon crystal. And so these grey balls here are silicon atoms connected by covalent bonds to each other, indicated by these grey tubes. And if we were to stack these unit cells one on top of the other in all 3 directions, we would build up a silicon crystal. And that silicon crystal is kind of, from that you would get this repeated pattern in all those directions. And that is what makes it a crystalline material as opposed to amorphous material where the atoms are randomly placed. And so if you can recall these wave functions that we talked about with individual atoms, if we have multiple atoms, we don't describe energy levels as wave functions anymore. We talk about the concept of a band structure. And a band structure evolves from a wave function and comes from considerations of multiple atoms close together in a crystal, such as in silicon. And band structure is also quite a deep concept to try and understand. But to put it simply in this diagram on the right here, where you see black lines, those are bands or energies where electrons can exist, and white space is where electrons cannot exist. And this grey band around 0 electron volts in energy is called the band gap. And in physics, what happens around the band gap determines the electronic properties of a material. So we're very interested in what happens in and around that band gap. Inside that band gap, usually there are no levels for electrons to exist. You can see there are no black curves inside that region, and therefore electrons cannot exist in there. But introducing the last kind of topic I want to talk about in this first part, what we can do is replace silicon atoms in the crystal with other atoms such as phosphorus, and this is called doping. And what doping does, illustrated here by these red balls, is that we can introduce electron energies into the band gap. Like I've shown here on the band structure in the right, there's now this red line, which is the dopant energy level, and that means that electrons can now exist within that band gap. And doping, this method of replacing silicon atoms with other atoms such as phosphorus, is going to be key to controlling how electrons exist, or how electrons exist at different energies within a crystal, and that will determine how electrons move around in that crystal, which is the topic of this talk. And so moving on, now that we have these basic ideas, I want to talk about tunnel junctions. So to begin with, what is tunneling? What is quantum tunneling? It's a concept you may or may not have heard of, but I think it helps to consider a real life analogy. So if you consider a lamp connected by a cord to a power socket, you turn it on and the light shines. And that's because electricity is a flow of electrons, which can flow to and from the lamp through the cord and do the work to produce the light. But if we were to quite unsafely cut the cord, we would expect the light to go off, because electrons can no longer get to the lamp to do that work. Electron tunneling comes into the mix because if we take this analogy and shrink it down to the atomic scale, or the scale of nanometers, which is 1/1000th of a meter, this analogy no longer holds, because if we analogously cut the cord in a device at this scale, electrons can still flow. If there's a barrier to electrons, there is a chance that they can tunnel through that barrier. And that is essentially what tunneling is, and is what I've been studying in my thesis. So if you were to look at an energy diagram here, this is analogous to the lamp that I showed in the slide before. On the left and right-hand side, you have the cord where the electrons can exist, and in the middle, you have the cut in the cord, which is a barrier to the electrons. And tunneling is simply the movement of an electron across this barrier, where if you were to take a real-life example, like a cut in the cord of a lamp, it should not be able to travel. And to study this, we make devices called tunnel junctions. And here is an image of a tunnel junction. And what this is showing is an image of a silicon crystal. And if you can see the length scale 10 nanometers there in the bottom left-hand corner, this is a very, very small scale, getting towards the scale of single atoms. These lighter regions here, surrounded by the white dashed lines, we've doped with phosphorus. So that means we've taken some of the silicon atoms there and replaced them with phosphorus atoms, which, as I showed before, inserts energy levels into the band gap of silicon, which means that electrons are available there and can move around. Whereas in these dark regions, which is just pure silicon, there are no energy levels there for electrons to occupy, which means that electrons cannot be there, which is what gives us this barrier in the left-hand energy level diagram that we see. And so how do we quantify electrons moving through this barrier, which is what we want to do? We want to look at how easily electrons can go through that barrier. What we do is that we measure the amount of current that can flow through these devices as a function of the bias or the voltage that we apply to them. And what we do is we get a curve like this, which is a IV or a source drain curve. And this curved feature here in the middle is what is the signature of tunneling. If there was no tunneling, there would just be a straight line. And to quantify how easily electrons can flow through this device, we take the zero bias gradient, R0, through the middle of this curve, which is basically the proportion of the change in voltage to the change in current at that point. And that quantifies how easily electrons can move through that junction. A higher resistance means that electrons find it a lot harder to flow through that barrier, lower resistance, vice versa. And so onto the results of our study, what we did is that we wanted to figure out how does the zero bias resistance R0, or how does the probability of an electron tunneling through that barrier change when you change the width and the length of that tunneling barrier. So we set the width to 7 nanometers and change the length. And you can see here on the left, we have an example of one of these tunnel junctions. We measure the resistance of that and plotted it as a function of length here on the right, which is this red dot. We then fabricated a couple more and plotted the resistance there as well. We then changed the width to 10 nanometers and changed the length, measured the resistance, and got these yellow points here. And then we repeated that once more for a width of 15 nanometers, which gives us the green points. So in this graph in the top right-hand corner here, we have the results of our study, which we'll now look at and analyze and try to extract some useful information from that. So first of all, if we look at the red dots here, we get a straight line, which because of the logarithmic scale on the y-axis, means that we have an exponential relationship between resistance and junction length, which is expected and predicted by theory. This plateau down the bottom here at roughly 20 kiloohms is also expected due to certain geometric limitations in our devices. But what was not expected was this large change in the position of the curve when we change the width from 7 nanometers to 10 nanometers, and which is what is essentially here 3 to 4 order of magnitude change in the resistance when we change the width by 3 nanometers. And this was quite significant because we had not seen this effect before. It's not in the literature, and we were very surprised to see it. And we are currently getting very close to publishing a paper about this, and we've been working with theorists who we collaborate with to try and explain it. And we're getting very close to a coherent explanation, which we hope to publish in the next few months. And so, how does our work fit into the field of quantum computing? So first of all, what is quantum computing? And I think it helps to start with the field of classical computing. Classical computing, it's the paradigm that powers the PCs and the phones that we use every day. And it's based on a device called a transistor, which is basically an on-off switch, which nowadays is made on the scale of 20 to 30 nanometers. And because these are so small, we can put hundreds of thousands of them in one chip, which fits in your phone or in your computer. And if you have many, many transistors together, you can make these circuits, and from these circuits, you can do these computations, which help us to solve problems, to communicate with each other, and many more things. Quantum computing, compared to that, is a new paradigm in terms of thinking about computations. It uses the reality of quantum mechanics at a very, very small scale to solve problems in a different way to classical computing. And the difference, or the main physical difference, is that in classical computing, you use a transistor as the basic element. In quantum computing, you use something called a qubit. And for qubits, what we use is a single phosphorus donor in a silicon crystal, like is shown here. And so these are the two differences. This is the main difference between classical computing and quantum computing. So you may be thinking, how does the tunneling through a tunnel junction relate to this? And the way it relates to it is through the surrounding architecture of the qubits. So qubits don't exist in isolation. You need to be able to do things to the qubits. You need to be able to know what they're doing. And tunnel junctions are a critical part of that. And so being able to know how easily electrons can tunnel through tunnel junctions, dependent on how wide or how long they are or where they're positioned, is of critical importance in building these machines. So to quickly summarize We looked at electrons in condensed matter. We looked at how dopants alter the allowed energy levels for electrons in silicon, which is how we control electrons in a silicon crystal. We looked at tunnel junctions. Using dopants, we can create devices where electrons tunnel through a barrier, analogous to cutting the cord of a lamp. And our research explains how the resistance of these devices changes and therefore how we can control the flow of electrons. And then I talked about quantum computing very briefly, about the basic parts of classical and quantum computing, and then how quantum computing is a new paradigm in computing, which our research facilitates by understanding how electrons move in a silicon crystal. Thank you very much for your attention, and I look forward to taking some questions.
Thank you, Matt. I think one of the most exciting and important evenings of the Royal Society's venture through a year is this particular evening. I get so excited when I see young people excited about science. And I spoke to all of our people tonight, and I did understand a couple, but Matt, I have to say that electrons moving through dark matter is beyond my very simple biomedical understandings. But let me get this thing started by saying that as someone who's very interested in the history of science, I've been fascinated by the way in which models have evolved and developed about how the atom exists. And I thought your talk was absolutely fantastic. I have to say I nearly understood it. I can't say I completely understood it, but I nearly did. And stop smiling, Ian. But what I've been fascinated in is the way our understanding of the atom has changed over time and how people working with these models have evolved things. And you started off with the, I think you left the poor old Rutherford atom out, but you got onto the Bohr. You got onto the Bohr atom, and then you moved on to the understanding how wave function changed. And I think this whole area of particles and waves is something that's just so revolutionary. And then you then moved on to band structure. And I'm just wondering, do you see the type of work you're doing changing the way we understand the basic atom shape?
Thank you very much for the question. Changing the way
we understand the atom.
Yeah. In a way, I think the big impact this work will have is not so much changing the way we see the atom, but changing the way, how do I say this? Changing the way that we interpret it in different materials, if that makes sense. So I don't think it will change the underlying approach that we take to solving these problems, which I kind of tried to reference there with, the electron wave function and different models of the atom. But I think it will give us a deeper understanding about how we understand, the effects of what that model, how that model looks in different materials, because those models of the atoms are generally applied. Well, they came about from studying hydrogen, which is the simplest atom there is, one proton, one proton, one neutron, one electron. And so applying that to heavier atoms like silicon and phosphorus is a very difficult question, is a very difficult process. And the theorists that I work with to help try and understand this, at the limits of the computational power we have today, it is still very difficult to understand what's going on with, with hundreds of these atoms. And when you're looking at these devices which contain thousands, tens of thousands, and possibly millions of atoms, it means that trying to understand that is a very difficult problem. So not exactly, it's not going to, I don't think so anyway, you never know, but I don't think it will affect the basic understanding, but it definitely affects how we apply that to different materials and atoms.
So Matt, you're saying that the current model allows you to do what you want to do rather than what you want to do demanding a change in the understanding of the atom.
Yes, exactly. Yes. So it's not going to demand a change, but we're in a sense applying that to a different question.
Right. Now, Stuart, you'll have to help me here. How do we get the other questions to Matt, who's dying to answer them?
If you just click on your Q&A function on your screen.
Oh, God, do I have one of that requires me to have electronic.
I can read them straight off.
Oh, wait a second. I can. Here we are. Susan Pong, who is a molecular lady, says, are there any examples of tunneling in biology? You've got to understand, Susan and I are basically biologists.
Mm-hmm. That is quite a coincidence, because I printed out a paper just about when I left work today for me to read tomorrow morning about that. Actually, it's a Nature paper from a couple of years ago. Yes, there are a lot, and I don't know a lot about it, to be honest. I've kind of perused the literature, and there's a lot out there. But I don't know the details of it. The paper that I was going to look at is basically about trying to understand, it's essentially isolating organic, I think, molecules or very small organic particles, and trying to drive a current through them and seeing how tunneling works there. But there's definitely applications of it in biology. But at this point, I haven't read too much into it. But I
won't feel so bad. Let me just see if there's not another one. I've got one, Matt. Why were you surprised that when you increased your gap, you increased resistance? Because it would seem to me that you would the greater the gap, the greater the challenge, and the greater the resistance for your electricity current.
Yeah. So it wasn't. It was changing the width. That was the thing. So changing the length increases the resistance, which was, I guess, on that plot that I showed. If you go further to the right on it, it goes up. But we were surprised because with the width, we were expecting quite a small change with the width. We weren't expecting that to be quite so influential a parameter. But then when we saw this, 3 or 4 orders of magnitude change in the resistance for 3 nanometers, that's why it was surprising.
So why do you think that's so?
That is a very good question, which we've been thinking about for a long time. There's lots of, there are a couple of answers to that. And most of them have to do with what I talked about on the second slide, which was band structure. So the band structure essentially, as I said, shows the energy levels that electrons can have, but it's very complex, and trying to work out the exact shape of certain parts of the band structure, and also how many levels there are, how it changes with different I guess, shapes of your device, is a very difficult question to answer. And so we don't know exactly why at the moment, although we're definitely honing in on I think, a probable answer at the moment. And it's very difficult to try and describe this, but it basically has to do with the fact that if an electron, depending on what direction the electron is traveling in within that crystal. So it's a 3D structure. It can travel in any direction it wants. Depending on which direction it's traveling in, it will see a different band structure, if that makes sense. It will there will be different energy levels it can occupy, and that is and that's basically what we think is causing this massive change.
So it's a very non-linear process.
Yes, exactly. So it's very kind of abrupt, and it kind of like it gives the electron a lot more energy levels to be in by changing that by a very small amount. We think that's what's happening, I'm having a meeting with my collaborators tomorrow to kind of flesh that out. But that's where we're going at the moment.
Terrific. Now, I've got another question for you, Matt, from someone who clearly knows more about this than I do, from Guy Lukes, who says, Matt, besides carbon, is there a possibility for an examination on materials such as carbon? It sounds like sort of reverting to the opposition, but
yeah,
to answer that.
So yes, there is. The reason, so probably just to preface this, the reason I'm looking at it in silicon is basically because that is the material, is because one, we already know a lot about that material because all the silicon, well, all the chips and the integrated circuits that make computers possible these days are made out of silicon. So there's been a lot of research over the past 50 years into it. So we already know a lot about it, and that's why our group are using it to try and make a quantum computer. So first of all, that's why we're using that. But with respect to carbon, there is also a lot of work going on with carbon. And I'm just trying to think of tunneling. The problem with carbon, the problem with carbon is that it's quite hard to dope it. So when I said before that you can replace silicon atoms with phosphorus and kind of inject those electron energy levels, it's difficult, or it's either difficult or you can't do it with carbon. But there are ways of investigating tunneling in carbon-based structures, such as there are things called nanotubes. There's graphene, which is quite well investigated for other reasons. But there are definitely investigations of tunneling, but they're not so much between, I guess, they're not, the technical terms, they're not planar, so it's not tunneling within the material. What they do with carbon is they kind of stack layers of different materials, like graphene layers. Exactly. They stack it and investigate tunneling in a different direction, if that makes sense. But there's definitely...
Let's move on to the last question because of time. Roger Reed, he seems to know more about this too. He does tell you it's a great talk, Matt, which we all agree with. Could changing the width allow different orientations of the dopant within the gap and therefore different pathways for the tunneling?
Yeah, that is a very insightful question because yes, that is definitely an issue, and that is something at first we thought was actually the reason behind this effect, because it's hard to explain without going quite deep into how exactly we fabricate these devices. But when you're doping, I think going back to, you remember there was that silicon crystal and I put those red balls where the phosphorus would go. You can control that to a certain extent, but there's always going to be an element of randomness in it, which I think you're referring to in your talk, because you do get these different orientations of the individual donors within it. But what we did is we modeled that with our collaborators and saw that while it does give you, it will give you some variation, it will, to the best of our understanding, will not give you an effect as large as the one we see. But that's definitely something that we've considered.
Terrific. Look, unfortunately, Matt, we could go on. When I hear talks like yours, Matt, it's the rare occasion I would love to have a one log IQ boost. But sadly, as a serious COVID-19 target, that's not going to happen. So I'm sorry, we need to move on. There are other questions, and I'm sure we'll fill them later. Now, the next talk is also, I think, very exciting. It's by Sajad Bazaz, who is going to talk about medical engineering and diagnostic tests, which he's the sort of guy I wished I had around to help me about 20 years ago. But unfortunately, he's a bit more modern, unfortunately for me. So thank you very much, Sajad.
Thanks for having me. I'm so pleased to present here at the Royal Society of New South Wales. My name is Sajad Razavi Bazaz from the School of Biomedical Engineering at UTS. and I would like to talk about my research activities, which is about 3D printed microengineered systems for life science research. So the first question is that what is microfluidics? Microfluidics is the study of systems that can process small quantities of fluids by using tiny channels having dimensions at the micro scale. Although in the nascent stage, microfluidics is rapidly emerging as a breakthrough technology that finds applications in diverse fields, ranging from biology and chemistry to information technology and optics. Without microfluidics, we face with bulky laboratory facilities, and each experiment takes so much time with huge amount of reagent consumption. On the other hand, microfluidics does all these tasks within a small channel that brings us the benefits of easy to use, portability, automation, multiplexing, polarization, and faster reaction time. However, their fabrication process is not rapid and they are not suitable for mass manufacturing. Therefore, we should find an alternative for the fabrication of microfluidic devices. 3D printing has the potential to significantly change the field of microfluidics. Developed in the early 1980s, 3D printing converts cat files into a physical object in a single process. Depends on the method of printing. Printing of a part usually takes less than 2 hours. It means that the brainstorming to performing the experiment can be carried out at the same day. The global 3D printing market size is estimated to be $10 billion in 2017 and is expected to reach $21 billion by 2021. Among all 3D printing methods, DLP or digital light processing has shown great promise for the fabrication of microfluidic devices. In microscale, fluids behave different than the macro scale. Therefore, it is essential to understand the underlying physics behind the microfluidic devices. To this end, through multiple studies, I have investigated the physics of fluids in microfluidics, as some of the examples are illustrated here. Through the knowledge that I have gained from the study of microfluidic physics, I was able to showcase the potential of microfluidic devices for life science research. Here, I bring some examples of these studies. So for the first study that I would like to present for you is the study with the collaboration of Cooper's company, the company that is famous for its beer in Australia. I'm sure all of you at least try one of the company's products. To provide a flawless product, breweries have to carry out a systematic quality control and test their beer for various contaminants. This is often challenging. Current routine microbiology quality control practices in the breweries are costly, time-consuming, and inefficient. Through the use of microfluidic devices, we have proposed a channel for improving the limit of detection of common beer spoilage bacteria in beer samples. Our channel is capable of high throughput and efficient separation of yeast from beer spoilage microorganisms. This is a clear example of the versatility of our microfluidic devices to be used in the industry. As the second example, I would like to talk about bioprocessing. Mesenchymal stem cells have shown great potential in treating multiple diseases and complications. To increase the production of mesenchymal stem cells to meet the clinical needs, microcarrier-based technology has gained considerable attention in clinical applications. However, the use of microcarriers has been hampered by a lack of proper system for harvesting cells from microcarriers. The current workflow of bioprocessing involves a series of expansion and processing steps, which are expensive, time-consuming, and often detrimental to the cells. Here, for the first time, I present a modular microfluidic system consisting of 2 micromixers, 1 spiral microfluidic device, and 1 zigzag channel to detach, separate, and concentrate mesenchymal stem cells from microcarriers. The microfluidic devices were produced with low-cost, simple, and rapid manufacturing process. Operating at high throughput, This microfluidic gadget is capable of detaching the cells fully from microcarrier with 5 minutes incubation time and 20 seconds processing time through the device. The cells passing through the device were shown to be viable and proliferative with their differentiation potential. More importantly, the therapeutic potential of the cells was well preserved. This system is sealed with no human intervention and has the potential to scale up and apply in the stem cell industry by multiplexing. Furthermore, this system is flexible and can potentially be modified to fit with any type of microcarrier and bioreactor for the manufacturing of various cell types and products. As the last example, I would like to talk about no IVF here. At this time, we coined it IV microwave. Unfortunately, 17% of all couples are infertile globally. In Australia, 1 in 20 babies, or about 1 in every classroom, born through IVF treatment. The IVF process includes the collection of eggs from a woman and sperm cells from a man. Sperm cells are processed to select the best cells for the job and introduced to the egg for fertilization. While the method of fertilizing the egg will differ, in all IVF treatments, we need to separate bad sperm from the good. Now here where it gets tricky, because although the number of IVF treatments is the highest it's ever been, the success rate have been stagnated to 33% per cycle, which means that the average couple needs to undergo 3 consecutive treatment cycles to get pregnant. Among all techniques suggested for sperm selection, the gold standard method is the density gradient centrifugation. Nevertheless, this method is time-consuming and applies huge mechanical forces that damage the DNA of sperm cells. Imagine providing a treatment but also risking the chance of success at the same time. And that's where we come in. To tackle these issues, we are using microfluidic technology to reframe the way we select sperm cells. Our system allows clinicians to use our disposable microfluidic devices at the size of a business card to sort sperm cells based on man's condition. Our device took only 15 minutes to complete the sperm selection process. It is relatively small and have the potential of parallel processing. The proposed system reduces human interactions, has less error, less labor, and less reliant on the skills variations of the technicians. We hope to see these disposable devices in clinics and help infertile couples to bring happiness in their life. This is our ultimate goal, actually. During my PhD studies, I was able to publish output of my research in prestigious journals, ranging from advanced materials to lab-on-a-chip and applied materials today, where 3 of them also got the journal cover. Moreover, I was able to patent one of my ideas following by submitting the other one to UTS. Furthermore, I achieved the most innovative prototype and People's Choice Award from UTS Accelerator 2019. A program helps students to build their ideas and shape it toward a successful product. Hence, as the output of my research, I'm pleased to say that together with my supervisor and my colleague, we built a startup named Microgenesis and working on that toward providing some disposable devices for better sperm selection. At the end, I would like to thank my supervisor, Dr. Majid Ebrahimi Valkiani, for all his support during my PhD journey. Also, I have to thank all members of Valkiani Lab and all academics in the School of Biomedical Engineering to provide facilities for our research. Thank you all for listening to my presentation. I hope my proposed 3D printed microengineered devices one day become a part of laboratories and help people toward a better life.
Thank you very much, Sajad. I mean, what exciting stuff. This is so familiar with many of us who have been struggling with these separation technologies. Let me get the questions rolling. When you started, this sounded very like going back to the old days of FACS, so fluorescein activated cell sorters. And I'm just wondering if you could just comment on how this technology has an advantage over that, well, I guess it's older technology now.
Okay, so thank you so much for your nice question, actually. There is a current project that we are doing is to make the fax systems more efficient. The problem with the fax is that still you have to do a specific binding to a target antibody, and these are the problems we have here. So that's how we want to make the fax process actually label-free. So the separation devices that I have shown you is based on the fact that we can go and separate the cell of target, but no need for any tagging or no need for any specification for them to select it. The fax or the max that people are using are very good, still using. In UTS, actually, we have many fax devices. Still, I am publishing those data. But the system needs to be much efficient, actually, from the current knowledge of separation systems that we have.
Right. There's a question here from Peter Bohm, who's a consummate physician. He wants to know if any of the IVF clinics are currently using the technique to identify motile sperm.
Oh, that's a nice question. So we start collaboration with clinics. Actually, we send our channels to some clinics. So they want to test them. They want to test them side by side with the conventional techniques and to see how the system goes. We are in the phase of clinical trial, as they call it.
Right. And finally, there's another question from Susan. She's very interested to see where you see your plans going after your PhD. And I think this is a really important question because one of the great challenges, I think, in our scientific communities is that we're basically helping over 10,000 people a year in Australia get PhDs. And I think we have a great responsibility to help and facilitate them moving into career structures. So we'd be very interested to hear, Sajad, how you see your future going from here.
Oh my God, that's a very challenging question, yes. So,
I think Susan might be offering you a job.
Actually, I have 3 or 4 months for finalizing my PhD. And during my PhD, due to the help of actually my supervisor and the team that we have developed so many ideas and so many devices here. So one thing that I would like to go is that I want to make my ideas change them. I want to translate my ideas to a product. This is my actual goal that I have. I know medical science, I know microengineering and mechanical engineering at the same time. So I want to combine this field to a product that this product can help people toward a better life. This is what I think I repeated 3 times in my talk. So my post-PhD plan is that to work on these ideas, make them as a device or some product, and then start going to fabricate and have these devices in every. My ultimate goal is that people go to chemist warehouse, similar to those diabetes tests that they can buy the product that we can produce.
We're running short of time, but I've just got to ask you this. The great challenge, one of the great challenges in biomedicine at the moment is COVID-19. What we're finding with the PCR testing, which is a standard testing that we all have when people stick things in your nose, is that we really can't tell if it's a viable virus or it's a fragment. And this has been, everyone's read in the paper about how the people who are in a hotel quarantine playing the Australian Open in Melbourne saying, oh, no, they're dead viruses. I don't really have the virus. Is your technology able to help us by taking cells from nasal scrapings, etc., and say there's a viable virus as opposed to just a fragment of RNA?
And that's a really amazing question. Actually, yes. So we have published one paper.
You've got a job in the future.
We got one paper published 2 months ago in the Journal of Diagnostics, which we talk about the current progress and the current help of point-of-care devices toward the COVID-19. So yes, what we are developing now is that to increase the sensitivity of these tests, we want to, for example, I think the current gold standard is beyond what we expect. That's why people with those dead fragments, they are still prone to the positive test, but that's true. We are increasing the, we are increasing the limit of detection here to make sure that we can get as many as viral cells in the sample. And if it says no, then we can say definitely that.
That's perfect. You've got a job for life. Sadly, I'd love to explore this, but we need to move on. Daniel Fox is going to talk about one of my very favourite topics. God, this is ridiculous, isn't it? On host-parasite relationships and the way in which he's examining the body's response to different pathogenic microbes. So, Dan, I know that we're all very interested to hear how you're going about this and where it's going.
Good evening, everyone. My name is Daniel Fox, and I'm a second-year PhD student at the John Curtin School of Medical Research at the Australian National University in Professor Seeming-Mann's lab. And I'm very proud and very excited to have been awarded a Royal Society of New South Wales scholarship. And I'm looking forward to presenting you and taking you through some of my research that I've been working on lately. So the topic of my presentation today is titled The Molecular Mechanisms of Inflammasome Activation by Enterotoxins of the Foodborne Pathogen Bacillus cereus. But firstly, before I begin, let me first introduce to you what an inflammasome actually is. Inflammasomes are essential components of the innate immune system, which is our body's first line of defense against invading pathogenic microbes. Inflammasomes are cytosolic innate immune signaling complexes, which assemble and activate upon detection of signals that signify a pathogen or of that of danger, such as tissue damage or mislocalized host cell components. In brief, upon detection of a signal, the inflammasome sends a protein, and there are many different kinds, which can detect many different kinds of stimuli, recruits a mediator protein, which can further recruit an effector protein. It is this effector protein which acts as a pair of molecular scissors, which has two primary functions. The first being is to cleave inactive cytokines into their active inflammatory forms and to elicit the inflammatory form of cell death known as pyroptosis. You may ask why our host cells are designed this way to program their own demise, but it makes sense if you consider an invading pathogen inside of a cell. This pathogen trips our inbuilt alarm system and sacrifices the compromised host cell to alert other cells nearby and to recruit other immune cells to further destroy the pathogen. Importantly, inflammasomes are also important in the regulation of many different kinds of inflammatory diseases, as well as cancer. A central question in our field, though, however, is how are extracellular bacterial ligands detected by cytosolic innate immune sensors? Or in other words, how do our signals outside host cells, how are they detected on the inside? An example of this has been recently shown by our lab. Here we found that the enterotoxin called HBl from Bacillus cereus can assemble on the membrane of macrophages, which is a type of immune cell, in a linear fashion to form a pore. This pore is permissive to the efflux of potassium ions, which is detected by the NLRP3 protein. This NLRP3 protein can then form what is known as an NLRP3 inflammasome complex. It is from here that the active NLRP3 inflammasome cleaves and processes inflammatory cytokines that generate inflammation, but also activates another protein that can form large pores itself to promote pyroptosis and the lytic form of cell death. These findings are important because Bacillus cereus is a clinically important and often neglected human foodborne pathogen. Typically, in immune-competent individuals, infection with Bacillus cereus will result in diarrhea and vomiting. However, in immune-compromised individuals, Bacillus cereus can cause a range of much more serious and deadly infections. Importantly, it is not notifiable and is self-limiting, which often makes it underreported. And of note is that in the US within the period of 1998 to 2008, 235 outbreaks of B. cereus was reported. Importantly, in addition to HBl, Bacillus cereus secretes many different kinds of toxins and virulence factors. So we wanted to delineate exactly how these other factors further interacted with the host innate immune system. Through a series of screening experiments, we were able to identify that the closely related non-hemolytic enterotoxin, or NHE, could indeed activate the NLRP3 inflammasome, like HBl. To examine this more closely, we utilized electron microscopy to look at macrophages at the single cell level. Here you can see the surface of the untreated macrophages have many different folds and ruffles, and the inside of the cell is very dense. However, when we treated these cells with the wild-type NHE toxin, we can see that this cell has been completely destroyed, and the cell morphology has completely collapsed these folds and ruffles. Moreover, inside the cell, you can see that it's much less dense, which indicates the cell has lost its cellular contents, and the cell nucleus has become quite dense as well, indicating pyroptotic cell death. However, when we treated these cells with a mutant toxin, which we generated, so this mutant toxin, we identified a region in the toxin, which we thought was important for cell killing. So we deleted this region and treated macrophages here. And you can see that they look very similar to the untreated macrophages, and they are quite dense, suggesting that they had not died. As such, we found that this putative transmembrane region in this toxin is required to mediate pyroptosis in macrophages. To further analyze whether this region was essential in the ability for the toxin to create pores in the host cell membrane, we constructed artificial liposomes which mimic the mammalian cell membrane. As you can see here, untreated liposomes possess no pores or holes in the membrane. Strikingly, however, liposomes treated with the wild-type toxin possess membrane pores. So this is what the toxin does. It creates pores in the membrane to create a hole which lyses the cell. Strikingly, this was the first time we ever visualized this toxin in this way. Moreover, the liposomes treated with the mutant toxin which we made did not possess any pores. This data therefore suggested that the putative transmembrane region in NHE is required for pore formation. We then further wanted to establish whether NHE could drive inflammation in the host in vivo. Here, using a septicemia model, we treated mice with the supernatant of Bacillus cereus culture, i.e. the broth from culture had no bacteria in it, but possessed any secreted products such as toxins which the bacteria had released. So with this supernatant, we either treated it with neutralizing antibodies to the HBl toxin, the NHE toxin, or both, or with an isotype control. And you can see here that only when we had neutralizing antibodies against both toxins did we see a significant decrease in IL-18 in the serum. So IL-18 is an inflammatory cytokine and is inflammasome associated. Therefore, this data suggested to us that NHE and HBl synergistically induced inflammation in the host. Finally, using the same septicemia model, we wanted to see if pharmacological inhibition of the NLRP3 inflammasome using a specific NLRP3 inhibitor, MCC950, could reduce NHE-induced inflammation in vivo. Here we found that indeed the inhibitor could very clearly reduce the levels of inflammation in the serum and peritoneal fluid in treated mice. This is important as during septicemia or sepsis, it is not necessarily the bacteria that kills patients, but the sheer shock of overt inflammation. We show here that this drug, MCC950, which is currently in clinical trials for other diseases, could potentially also be of use in septicemia. Therefore, therapeutic modulation of the NLRP3 inflammasome dampens NHE-induced inflammation in the host. Overall, to conclude, we found that both the HBl and NHE enterotoxins from Bacillus cereus can be detected by the NLRP3 inflammasome to drive the processing of inflammation pyroptosis. So to sum, we found that multiple virulence factors from the same pathogen can be exploited for sensing by a single inflammasome, and that these results give novel insight into the innate immune response against Bacillus cereus, a clinically important and neglected human foodborne pathogen. Finally, I would like to acknowledge my supervisor, Professor Seeming Man, for his constant support and guidance during this project, the NHMRC, ANU, and JCSMR for providing financial support, the other members of the MAN Lab for their help, in particular Anacrity Martha, and all other internal and external collaborators for that input. In addition, I would also like to sincerely thank the Royal Society of New South Wales for their recognition of my research with this award, and I would like to thank all of you for listening. I'm more than happy to take any questions you may have in the Q&A session coming up. Thank you.
Daniel, I'll just check. There'll be a lot of questions here, not just, they're all coming. I'll get the ball rolling a little. One of the things that I think is incredibly important for people like yourself, who I assume have a science background, is that this extraordinarily interesting, really exciting research you're doing gets translated into a human picture. And it struck me that along the way, you were looking at deleted sequences in, I think it was a transmembrane protein that prevented the pore formation, which struck me as being a very good way to go towards a vaccine production, perhaps in other areas which have greater human significance in terms of their infectivity and the impact of disease. Have you thought along these lines? Are there, in fact, parallel host-parasite relationships which are of more relevance to day-to-day human disease that have this same type of mechanism where you could delete a transmembrane sequence and create a very effective vaccine?
That's a really good question. So we looked at that. So the point of looking at the transmembrane region in particular was, sure, if you delete a transmembrane region, it's likely not going to bind to a cell membrane and therefore not be cytotoxic, which you could use for a vaccine. So this isn't out of reach. So things like diphtheria and tetanus use the same approach. A similar approach here for this toxin. However, B cereus is very complex because it can secrete a lot of virulence factors. So the two that I touched on were the HBl and the NHE toxins. These are the main drivers of disease in humans. But what's important is we did the same thing for the HBl toxin. So we found the putative transmembrane domains and we deleted them and we found a completely opposite effect. So in this toxin, what happened was it was able to bind better to cells. So whilst it is in theory a good idea to create a vaccine with mutant toxoids, we really need to fully understand how all of these toxins interact with the immune system first and make sure that each of these mutant toxoids will work and not make the disease worse, if that makes sense. So we really need to dig into the nitty-gritty.
Right. I think we're still waiting for all these powerful questions. Let me go back to, while we're waiting, let me go back to right at the beginning. What you talked about to me sounded a very exciting ecological system where you've got a community of cells where one cell's prepared to give up its life to save the others. And I just wonder if you could expand a little bit on that. Is that a concept that I have taken too far, or is that an idea that is well established in your folklore?
It's not too far at all. So this is, I guess, why in terms of, the drug that I came in with at the end, that was to dampen the immune response. So if you consider why we would want these cells to die in the first place, in an immune competent person, if you get a localized infection such as Bacillus cereus, you want the cell to detect that, realize something is wrong, release inflammation to bring other cells to the site of infection to try and clear the infection, and hopefully it will go away. However, what happens, say with sepsis, is you don't have a control over the system. And some people have autoinflammatory conditions where they have genetic mutations in the regulators for the system. So if you remove the brake, you're going to just keep getting a massive amount of inflammation, and you're going to get heaps of cell death happening, which is why in sepsis, you want to dampen the system down, because it's not the bacteria that's killing you, it's your own immune response just completely going haywire. So this system is very, it's a fine balance, and it's why we really need to work out how we can control it, but also how understanding the mechanisms of which it turns on. So it can be both, it's a double-edged sword, I guess.
Yeah, it's very, very interesting concept. I mean, I guess it's the fine tuning of like a servo system. It's very interesting. We'll get our people in electrons to come along and give you a hand there, Dan. But sadly, time is moving, as do electrons and as do macrophages. But we are moving to the real whole person now. And we're moving to a presentation by Philippa Specker, who is going to talk about post-traumatic damage in a very important group of people who are very traumatized by translocation across borders. So, Philippa, we're very, very interested to hear. So we're really talking now about whole people psychology and the damage that can occur with national translocation. Thank you very much, Philippa.
Hi everyone, my name is Philippa Specker and I'm a PhD student at UNSW, and it's a real honour to be here. Today I'll be discussing my research, which investigates the mechanistic role of emotion regulation flexibility in the development of post-traumatic stress disorder among refugees. So let's get started. Okay. So to begin with, it's important to acknowledge the humanitarian crisis that's currently occurring. There are an estimated 80 million refugees worldwide, and unfortunately this number is growing. The refugee experience is fraught with trauma and stress. In the home country, this includes things like war exposure, torture, and imprisonment. When fleeing, refugees often experience further life-threatening traumas and family separation. And once in a new country, refugees have to contend with an extraordinary amount of stress due to things like language barriers, financial strife, and visa insecurity. Unsurprisingly then, these experiences place refugees at a greatly increased risk for developing psychological disorders. In particular, refugees represent one of the largest at-risk groups for the development of PTSD, with as many as 1 in 3 suffering from this disorder. And herein lies the global challenge to public health. Although psychological treatments for PTSD exist, they weren't designed with refugee trauma in mind. They weren't designed to address the psychological complexities of refugee experiences. And so they just aren't as effective for refugees. Although we know that these types of refugee experiences and traumas lead to a high likelihood of PTSD, unfortunately, we know very little about the unique psychological mechanisms that might cause the development of PTSD following refugee trauma. And if we're going to improve existing PTSD treatments for refugees, we really need this information. This is where my PhD comes in. Although not well researched yet, one mechanism that looks particularly promising in explaining this relationship is emotion regulation. This is because emerging research has found that refugees are especially likely to experience emotion regulation difficulties, owing to their experiences of interpersonal trauma, displacement, and post-migration stresses. Now, emotion regulation just refers to our capacity to monitor, evaluate, and modify emotional responses to meet the demands of a situation. Without even realizing it, we all use strategies to manage our emotions. For example, sometimes we might reappraise a situation to help us feel less negative. Like I might tell myself that the rain is actually a great opportunity for my garden to get watered. Or instead, I might actively suppress my emotions by trying to push away my frustration and avoid thinking about the rain altogether. I focus on these two strategies because research suggests that people with PTSD tend to use a lot of emotional suppression and very little cognitive reappraisal in their daily lives. But the problem is that most existing research tends to look at the impact of these strategies in isolation. But in the real world, we tend to use a variety of strategies throughout our day. To better capture real-world emotion regulation behaviour, it's been argued that we need to consider someone's emotion regulation flexibility. Now, this just refers to how flexible someone is in the way they select and use emotion regulation strategies. Someone with high emotion regulation flexibility will have a large repertoire of emotion regulation strategies at their disposal, and they'll be good at selecting the appropriate strategy or strategies across different situations. This begs the question, do people with lower ER flexibility have poorer psychological functioning? And if so, can ER flexibility be taught to them? When I started my PhD, these questions had not been explored in the context of any mental illness, let alone among refugees with PTSD. So I thought I'd do just that. My research seeks to answer two questions. First, is emotion regulation flexibility implicated in the development of PTSD in refugees? And if so, can ER flexibility skills training reduce PTSD symptomology? To answer this first question, I explored emotion regulation profiles of refugees in both naturalistic and experimental settings and investigated how different profiles were associated with psychopathology. Now, profiles just means that I looked at each individual's pattern of emotion regulation strategy use across multiple strategies. So I looked at their full repertoire in one go, rather than one strategy at a time. To do this, we use a statistical technique called latent profile analysis, which allows us to group individuals into latent profiles or homogeneous subgroups according to common use of certain emotion regulation strategies. This allows us to compare how refugees with different levels of ER flexibility might differ in their psychological functioning. And the results were really interesting. So first, in our first study, we asked a culturally diverse group of adult refugees to complete questionnaires in their native languages. And these questionnaires assessed, firstly, how much they routinely used two key emotion regulation strategies to manage their stress in their daily lives. And we focused on reappraisal and suppression in particular. And we also assessed each participant's level of PTSD symptomology. With this information, we were able to look at each person's emotion regulation repertoire across the two strategies, and we found three predominant groups. Just to orient you to this graph, along the bottom, the x-axis indicate the items assessing the two emotion regulation strategies. The first 5 items measure suppression use, and the last 6 items measure reappraisal use. Along the y-axis, we have item probabilities, where a higher number indicates a higher probability that people in that group endorse that item or use that strategy. And what we found was that the bottom group, the green group, which characterised individuals with low emotion regulation flexibility because they just used one strategy. In this case, they just used suppression all the time. They actually had significantly more PTSD symptoms. This was the first piece of research to suggest that lower emotion regulation flexibility is associated with greater PTSD among refugees. Then we did a lab-based study and found similar results. This time, a culturally diverse sample of refugees came into the lab and we assessed their PTSD symptom severity. Following this, participants viewed trauma-salient images for 5 minutes. These images were quite unpleasant. They served as a temporary emotional stressor. And immediately following this, participants rated how negative the images made them feel, and they completed a short questionnaire to assess how much they used reappraisal or suppression to manage their negative emotions when they were viewing the images. And this time we found two groups. Individuals that demonstrated high ER flexibility in response to the stressor, the red group, and this is because they used high levels of both strategies. And then individuals who showed comparatively lower emotion regulation flexibility, the green group, and this is because they made less use of both strategies. And what we found was that individuals in the low ER flexibility group had a higher PTSD symptom severity. On top of that, this group also reported feeling more negative after viewing the images than individuals who had used a more flexible approach to manage their emotions. This suggests that refugees who exhibit lower ER flexibility in response to stress are less able to manage their negative emotions in the moment and are more likely to have PTSD. The findings across these two studies are really exciting because this is the first time anyone's been able to demonstrate a connection between lower emotion regulation flexibility and greater PTSD symptomology. So the answer to my first research question is yes, we think so. So our next step is to test for a causal link between ER flexibility and PTSD. We will do this in the lab to test whether providing training in ER flexibility prior to viewing trauma-related images reduces symptoms associated with PTSD, things like intrusive memories. If it does, then that would be really exciting preliminary evidence suggesting that ER flexibility is a viable clinical target for PTSD. This will then become an important building block in the development of more effective treatments for refugees with PTSD. I plan to conduct this study virtually this year, and I can't wait to see the results. So I do want to end by saying a huge thank you. Firstly, I want to thank all the refugees who take part in this important research, and also to my formidable PhD supervisor, Professor Angela Nickerson. And finally, I'd like to thank all of you. Thank you so much to the Royal Society of New South Wales for this award and recognition.
Thank you very, very much, Philippa. What a wonderful way to end this. I was just thinking we start with electrons and we end with real people. You know, a few of us here tonight, when we did psychiatry, the little we did, there was no such thing as post-traumatic syndrome. It just didn't exist. And to see that now we're looking at moving towards understanding some of the psychological mechanisms behind it, I think is a hugely exciting advance, because that will lead to some form of therapy. Now, Peter Bohm has a question, and he agreed with me that you presented very interesting experimental results of a group of refugees. Would you tell us a little bit about the sample? And I think this is a good question. Peter wants to know whether the samples were selected, and I'll just extend that. I mean, were there differences between different countries? Was there a sex difference? Can you give us a little bit more insight into perhaps the correlation of the samples and the outcomes?
Yeah, thank you for that question, Peter, and for your comments as well, Robert. So all of the participants in all of our studies come to us from various channels. All of the participants who take part are refugees that are resettled in Australia. I'm really lucky. I work in the context of a larger research lab called the Refugee Trauma and Recovery Program. And this kind of research lab and clinical program has been around for a number of years and is heavily involved in the kind of refugee service provider kind of sector, as well as treatment provision and a whole number of different organisations that they work with incredibly collaboratively, like Red Cross and Settlement Services International, which are the key bodies that refugees interact with as soon as they arrive in Australia. And so because we have this, really fruitful research collaboration and partnership, a lot of our studies are advertised in those areas, and refugees will routinely self-refer to take part in research and to also access affordable treatment for PTSD through our program. So that's how they come to us. And we now have a database of over 5,000 refugees resettled in Australia that want to take part in research or hear updates on the work that we're doing, which is incredibly exciting. In terms of whether there might be cultural differences in the effects or sex differences, that's a really great question, and one I've indeed asked of the data. The kind of analysis that we did in these two studies that I presented on, in order to ask questions when you come down to sex differences or cultural differences, we tend to need a much larger sample in order to have the statistical power to be really confident that we can actually detect differences between those groups, because it would require cutting our sample again into smaller subgroups. So it's definitely a question that I've got at the back of my mind, and it's hard to know whether that would be the case because there is so little research out there that's conducted with refugees, but it's definitely something we'll circle back to. I do think, though, particularly in the context of emotion regulation research, it's really important to consider cultural differences, because we know that the way that we experience and therefore regulate our emotions can sometimes be mediated by our cultural experiences and beliefs around emotions and strategies. So it's probably incredibly relevant, and it's something I continue to focus on as I start to plan this next study. So in our next study, all of the participants will be from the same cultural group in order to test for that effect.
Really got waiting for all these extra fantastic questions that are in the, but I'll just ask, it seems to me that you're onto something, Philippa. I'm wondering if there's a way in which you can use your technologies for sort of very early assessment of people coming to our wonderful country, so that you can actually put in place programs at a very early stage. And the other quick question I'll ask is, how do you know that your various answers are not basically cultural identifiers? In other words, do you look at control groups of people who do not have post-traumatic syndrome, that maybe some of these technologies are part of either a cultural or some other non, if you like, pathologic identity?
Yeah. These are great questions. I should be writing them down as you mention them. So I think the first thing is the question of a control group is so important in scientific research, and it's one that's often quite difficult to think of what's the appropriate control group when you're working with refugees who have PTSD. Is it refugees who don't have PTSD? Is it people who are immigrants but they're not refugees because they haven't experienced XYZ experiences? So it becomes quite difficult. Something that is quite handy in the way that at least the two studies I presented, I approach in terms of sampling, is this is more or less a kind of representative sample of, refugees who have more or less recently arrived in Australia in the last decade. Which means that what we have in the sample is a number of refugees who have PTSD, but also a number of refugees who might have had similar experiences, but for whatever reason don't have PTSD. And that's where it gets really interesting from my perspective, because how is it that someone who's experienced similar traumatic events like war exposure can not develop PTSD? Because these are kind of extraordinary events, and yet there seems to be about 1 in 3 refugees that have this resilience and don't go on to show significant symptoms of PTSD or other disorders. And so we can actually use them in our study as kind of a control group to see, okay, similar experiences but different outcome. Maybe there's a different mechanism that's underlying this that can explain these divergent pathways. And so in the research that I presented on today, seeing there was kind of a high group and the low group almost kind of starts to demonstrate this, this possibility that there's, I guess, two divergent outcomes within the context of emotion regulation, and one might lead to you developing PTSD and the other might not. So that was kind of a really interesting thing to see in our sample, that it kind of fell out that way, which suggests that perhaps this was kind of a natural control group that we used within the sample. I'm not sure if that answered your question there, Robert.
No, I just think it's so exciting. Look, the great sadness is that we've come to a time where we've got to sort of sign off, but my very privileged role is to thank all of you tonight, to thank Matt, Sajad, Daniel, and Philippa. And, you know, when I look at what you guys are doing, we really should have a big dinner where we get you together, because I can see that what we've done tonight is that we've gone from an atom that I struggle with to cells that we can now identify with a very, very interesting methodology. We can look at the cells and their relationship to what's going on around it. And we eventually put all of these things together and get a whole person. Now, Philippa, what I think you should be demanding of these guys is they help you identify the gene that determines these various changes. I have a daughter who's a speech pathologist, and I've tried this on her many, many times with no success at all, because a lot of the technologies you're talking about are not too different to some of the ones she uses in autistic disease. But that's what's going to happen, you know, Philippa. In your young lifetime, you're going to be working with these very clever people that you're surrounded by, and you're going to come up with a genetic control, and we're going to have gene transfers and all sorts of wonderful things. Pretty scary stuff, isn't it? But my job is just to thank you so much for giving us new insights, exciting opportunities to view what is going on in the world around us and where our life is going. So thank you so much, everybody. It's a privilege for us, and we appreciate it. Thank you.
Yeah, well, thank you very much, ladies and gentlemen. That concludes the evening. Thank you for your attendance. It's been lovely to have you. We'll be back next month, of course, for our OGM. And if you want to hang around, we'll play some music and show some information about the upcoming OGMs and lectures for the year ahead. Thank you all once again.